आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सोल्यूशन पाओ। डाउनलोड नाउ।
The question here is that DNA being the hydrophilic cannot pass through the cell membrane of a host cell. Explain how does the recombinant DNA get introduced into the host cell to transform the latter here, right? So let's see the answer now. So if we talk about the cell membrane, so cell membrane is the outer covering of the cell here, and it has the presence of the lipid layer, and it is the layer which only allows the movement of molecules in and out of the cell here, right? Now this lipid layer only allows the hydrophobic molecules to pass, right? It only allows the hydrophobic molecules to pass through it, right? But here DNA is hydrophilic in nature, so that is why it cannot pass, because only the hydrophobic molecules they are allowed here, right? Now we have to tell that how the recombinant DNA can be introduced into the host cell here, right? So we have the methods here with the help of which the DNA can be introduced here, right? So the first is we have the gene gun, right? So the first is the gene gun here. So with the help of the gene gun, or we could call it as a biolistic also, right? So what happened is there, so the cells in which the gene has to be inserted or DNA has to be inserted, they are bombarded, right? So the cells, they are bombarded with the high velocity microparticles, right? So the high velocity microparticles, they are bombarded right towards the cell here, and these microparticles, they are of gold or tungsten, and these gold and tungsten particles, they are coated with DNA here, right? So along with this particle, the DNA also enters the cell here, right? This is the first method. The second method that can be used is the heat shock method here, right? So what is heat shock method here? So what happens is, in this method the DNA is treated with the specific concentration of the divalent cation, right? So DNA is associated with the divalent cation, such as we have the example of the calcium here, right? And with this, what happens is there is the increase in efficiency, right? There is increase in the efficiency of the DNA, with which the DNA, it enters the bacterium, right? DNA enters the bacterium, and that entry is through the pores here, right? The cell wall has the pores here, right? So the DNA enters the bacterium through the pores of cell wall here. So this is the second method.
क्लास 6 से 12 से लेके नीट, IIT, जेईई मेंस और एडवांस के लेवल तक 10 मिलियन से ज्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।